So from last class, I had a Socratic quiz where I asked, what did you learn that you need to know more about? And I'm just going to quickly summarize some of what you said. These are a number of comments that I got categorized into these 6 different categories. We're going to talk about all of this later in the semester, so I'm glad you're thinking about these sorts of topics. We're not quite there yet. We'll get there. Now in red, which is kind of hard to see when it's projected, especially if you're red-green colorblind like me, is our topics that we're going to cover today and probably into Friday. So we're going to be on this chapter, chromosome structure, at least today, maybe partway into Friday. We will definitely do more practice on karyotype versus karyogram, which was the #1 request from all of you, and then how you get from a karyogram, the photograph of metaphase chromosomes, to the organized version, the karyotype, which is the numerical description. So that process apparently is not quite clear to everybody, which is fine. We've got plenty of time to do this. Defining ploidy, that is how you know if an organism is N, 2N, 4N, what the hell N means. And then I got some really interesting extension sort of questions, like, well, what does this all mean? Of course, you should want to know why this is relevant to biology or to you particularly. What does it matter? Are there differences in chromosome number between species? Does it matter that some chromosomes are metacentric, submetacentric, acrocentric, telocentric? So we're going to talk about all of those topics. And what's left in black then is, so the significance of centricity, here's the answer to that question. As far as any geneticist knows, there's no difference to the chromosome, whether it's metacentric, acrocentric, telocentric, submetacentric. We just use those terms to easily communicate with other geneticists what sorts of chromosomes an organism has. No functional significance in terms of how the chromosome works. It doesn't matter where its centromere is as long as the centromere exists. Okay, so that's the short version of that question, the answer to that question. And one question, is there a way to know which chromosomes come from the mom and which come from the dad when you look at a karyogram or a metaphase spread? No. You look at the chromosomes, we know that in humans, there are 2 copies of chromosome 1, 2 of chromosome 2, and so forth. But just by looking at them, you don't know which ones came from which parents. What you would have to know, which this person was pointing out, I think, is that you would have to have the DNA of your parents to know which of your chromosomes came from which of them. So you'd have to have that reference. Any questions about any of that? Any follow-ups? Yeah.
Wouldn't centricity, where the centromere is on the chromosome, affect crossing over?
Where the centromere exists does affect where crossing over happens. That's a minor effect, but not in terms of the function of the chromosome. But you're absolutely right. Yep, and we'll get to that later in the semester when we talk more about crossing over and meiosis.
Okay, thank you.
Please bring that up again. And then today what we're going to get to also a few things. These are the main points. Somebody asked for electron microscopy images of chromosomes, so here's one. The human, the X and the Y chromosomes. Anybody know which is which? What's on the left?
The Y chromosome.
Yeah, the Y chromosome. And it's not just because it looks like a Y, although it kind of does. That's actually not why the chromosomes are called X and Y. We'll talk about that when we talk about sex chromosomes. But the male chromosome, the Y, which determines maleness, is literally falling apart. It has been over the last 300 million years or so. And so we're destined to extinction at some point, probably. But you can't live without us because women, as far as I can tell at this point, can't reproduce by themselves. So if males go extinct, then the whole human race goes extinct. So there's sort of a balance there. We'll talk about it when we talk about sex chromosomes. We'll practice doing some more literature searches today. We'll definitely get to the restriction enzymes by the end of class today. I know a lot of you were maybe a little bit worried that we hadn't gotten there yet. It was part of this chapter. We've still got plenty of time. So we're not moving on yet. We're going to get there. And then isochores. There were a number of questions. Again, we haven't gotten to that part of this topic yet. But the short answer to that is what's the relationship between isochores and banding? Chromosome banding we did talk about last class. The point of banding, again, just to make a long story short, we'll talk about banding more a little bit, is banding tells us which chromosomes are identical, the same as each other. So that should have been a takeaway you got from last. So isochores. This was part of one of the reading materials from the first chapter, chromosome structure, which we're still on. And one of the questions is, what is an isochore, and what does it have to do with banding structure? So this supplemental reading, which was linked in the textbook, the course manual, is important. There's a relationship between isochores and chromosome banding. This is an isochore plot down here on the bottom. And I've just blown up. So which chromosome is this? This is human chromosome 14 in this graph. So I've taken an image of human chromosome 14, a banded image, and it's scaled now so that the width or the length of this chromosome is the same as the length of the plot. So isochores, very simply, are loosely defined regions of chromosomes that have either more G's and C's or more A's and T's. That's it. And if you look at this graph, first of all, the next couple of slides are going to ask a few questions about what are the axes and so forth. So I suppose I can move to those. So here are 3 different plots, top, middle, bottom, all from this paper. Okay, first, in analyzing any sort of graphic, whether you've looked at this already, I hope you have, but if not, oh geez, a lot of data. So to make sense of the data, first think about the axes. What's the x-axis in this case? What's being measured on the x-axis? This one. So coordinates, okay, what sort of coordinates? The units, of course, are always in parentheses. So MB, mega base pairs, or million base pairs. So sometimes you'll see MB, sometimes it will be MBP. It's million base pairs. And that's because, as we talked about last time, chromosomes are so huge that it doesn't make sense to plot just base pairs because the numbers, you wouldn't be able to put 24. You'd put 24 followed by 6 zeros, and then when you put the numbers next to each other on the graph, they'd all just be running into each other. So we just compress the numbers by measuring in millions of base pairs instead of individual base pairs. So how long is this chromosome? They're not even showing us the first 10 million base pairs. 0 to 10 would be to the left. It's not even shown. So we're looking at between 10 and 24 million base pairs worth of genome sequence. Everybody okay with that? So nucleotides, just units of genetic distance, physical genetic distance on the x-axis. And then what's the y-axis?
Percent GC.
Percent GC, 20 kb windows. So what that means is that these authors look at 20,000 base pairs at a time, which is a really small amount on the units of measuring millions of base pairs here. They look at 10 or 20,000, 20 kb, 20 kilobases, 20,000 base pairs at a time. So they take a contiguous stretch of 20,000 nucleotides of sequence, say from 0 to 20,000, and they say, what is the percent of those 20,000 nucleotides that are G's and C's? So if I wrote this sequence, what's the percent GC? 5 nucleotides.
40%.
So 2 out of the 5 are G's or C's. So 2 divided by 5, 40%. So that's essentially, that's 40% GC. GCATA, 40% of those 5 letters are G's or C's, either one. So that's what these scientists are doing. They're just doing it on huge stretches of DNA, 20,000 nucleotides at a time. They say, what's in this part of this chromosome, what's the percent of the nucleotides that are G's and C's? And then they measure that at every point along this graph. So they measure it here, they measure it there, 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 there. They keep moving down the chromosome, 20,000 base pairs at a time, calculating this number. Okay, so what does it mean? So let's look at human on the top. Oh, I guess I should have asked this question first. What are the differences between the 3 graphs? Why are there 3 different graphs?
Variability kind of.
So what are the titles on these
They're different.
plots? So they're different, first they're different species, but they're also different chromosomes. It turns out that this is an unfortunate thing about genetics, is different people work on different organisms and sometimes they don't talk to each other. Sometimes people have worked on different organisms at different points in time. And so whoever it was that studied chickens, they looked at the chromosomes of the chickens and they said, you, chromosome that I'm looking at right now, are going to be called chromosome 5. And unfortunately, when human geneticists were studying human chromosomes, it turns out that that same chromosome in chickens, the human geneticists called chromosome 14. So these are the same chromosomes in 3 different species. They're just numbered differently because the people that work on those 3 different organisms happen to choose different numbers for those chromosomes. It's unfortunate, but there's no perfect way to go about doing that. So human on the top, chicken in the middle, and Anolis carolinensis, it's a lizard, on the bottom. This paper was about the Anolis lizards. And that's why there are 3 graphs. So they're measuring this, looking at the same chromosome in 3 different species and plotting GC content, percent GC across this one part of this one chromosome. And then for those of you that are in the back that maybe can't see way down here on the screen, hopefully you're looking at your devices, what it says down at the bottom, this is always the question you should ask every time you look at a graph. There's something, scientists are bad at this, sorry, apologies in advance. There's something about the comparison of these 3 plots that you're supposed to see that's really important to these authors anyway. Hopefully it's important to you also. They're trying to make a point by comparing directly that, and that. So what's the difference between the 3? Is there a big trend? Is there something that's unique about one of those 3 or 2 of those 3 graphs?
The human chromosome 14 and chromosome 5 look similar. They're graphs.
Okay, so chromosome 14 in humans, on average, sort of does that sort of thing. If I drew a trend line through all the data points, there's a lot of variation. Where's more of the variation? If you look from one set of 20,000 base pairs to the immediately adjacent and keep going like this, where there's a lot of where is there more variation?
On the 2 ends.
Yeah, it seems like the line, this plot goes up and down a lot more drastically at the ends of the chromosome, and maybe there's still variation in the middle, but it's just smaller peaks of variation from one window of nucleotides to the next. Okay, so does the human look the same or different than the chicken?
Similar.
Yeah, it's similar. It's lower GC in the middle of the chromosome, higher maybe a little bit on the ends. And then the point. When you contrast both of those to the lizard, what happens?
The lizard doesn't have a lot of variability throughout the entire.
The calculation of percent GC is almost the same. There's some variation still. There's some big peaks here in the middle of the chromosome, but they're really narrow peaks. But on average, that Anolis carolinensis GC percentage plot, which is called an isochore plot, is pretty flat. So the point of this paper was to show that exactly what we just said. There's no variation from end to end of a chromosome in these lizards in the number of G's and C's versus the number of A's and T's. But this is true in humans and chickens. The takeaway here, there are 2 of them. One is that depending on where you look on a chromosome in humans and in many other species, depending on which part of the chromosome you're looking at, near the ends, which are also called the telomeres, the ends of the chromosomes. In humans, there's often more G's and C's towards the telomeres, and the centers of the chromosomes tend to be more rich in what nucleotides then? Have more A and T. So if there's low GC, that's the y-axis, right? We're measuring how many G's and C's. So if there aren't G's and C's, then there must be A's and T's. So low GC percentage is the same thing as high numbers of A's and T's relative to G's and C's. This is surprising to scientists. They sort of expected that, well, there are only 4 letters of the alphabet, so on average, if you look at any part of any chromosome, there should be about 25% A, 25% T, 25% G, 25% C. That's true in this one lizard species on this one part of this one chromosome, but it's not true in chickens and humans. Nucleotide composition varies depending on which part of which chromosome you're looking at. It's not just random. So these regions, finally, these regions that have lots of G's and C's are the things that are called isochores, just to reiterate that. So an isochore, again, is a region of a chromosome that's defined by this sort of plot that has more G's and C's or more A's and T's than expected, than average, in other words. So these 2 parts, is this green?
Yes.
Okay, thanks. The 2 parts that I circled in green are isochores. And this part in the middle would also be a separate isochore. So we got high GC, low GC, high GC from left to right. There's no formal definition of numerically what constitutes an isochore. So you're free to have a little bit of artistic liberty about if I ask you to define isochores based on this sort of a plot. Yeah.
So just to summarize, an isochore is when it has more or less than expected.
Then average, let's say. Yeah. More GCs than average or more ATs than average. And that's why this lizard genome sequence is a great contrast between humans, chickens, and lizards, is that this is what you'd expect if there were no isochores. There's no variation from one end of the chromosome to the other in average numbers of G's and C's. So what I didn't mention a few slides back was I showed a picture of a banded chromosome. So I'm just going to draw in an ideogram of a banded chromosome. The reason that chromosomes have stripes, the answer to this long-standing question now, who was thinking about it, is depending on the chemical you treat the chromosomes with. Remember, that's how we get chromosomes to have bands. We have to stain the chromosomes with a particular chemical. There are a number of different chemicals. That's not important. There are a number of different ways to elicit bands from chromosomes. But one type of chemical only stains when there are G-heavy isochores. So if we plotted on this one, just like we were looking at percent GC, in that plot, every time there's an increase in GC content, that's where the chromosome would stain darkly for this particular dye, generic dye that I'm talking about. So there's something about high numbers of G's and C's in a very compact region of a chromosome that with stains that cytogeneticists apply, people that study chromosomes, those regions of those chromosomes stain more darkly when they have lots of G's and C's, and that's how we get chromosome banding. And those banding patterns are unique to every chromosome because no 2 chromosomes are alike in exactly where they have dense areas of G's and C's. Time to get to the restriction ends of endonucleases. So one of the questions that I had asked in this video was, what enzyme and from what bacterium cuts this particular sequence? GC, GG, CC, GC. And then the second question was sort of the advanced question. If we gave you this DNA sequence, what mutation would make a restriction site for a particular, that is the DNA sequence that this enzyme, HIND3, recognizes? So restriction enzymes, what do they do? These are proteins, enzymes, proteins. Do you know what a restriction enzyme's job is, what its role is, what it does inside the cell?
Doesn't it, look for mistakes in the DNA, or is that part of DNA synthesis?
That's a different enzyme, but it does have to do with DNA. Restriction enzymes, there are a bunch of different names of restriction enzymes, a bunch of different proteins. Each of those proteins has a particular DNA sequence that it recognizes. And that protein will bind to that DNA sequence, whatever the sequence it is, and it will cut the DNA where it binds that particular DNA sequence. So, for example, there's this enzyme that cuts this 8 nucleotide sequence. This protein floats around in your nucleus if you have this protein. These come from bacteria, by the way, not from humans, so you don't actually physically have them in your nuclei. These bacteria have these enzymes that float around in the cytoplasm, and every time this enzyme sees that DNA sequence in DNA, that protein will bind to it. And so let's just be really explicit and put in the complementary nucleotides here. Base pairing. C's pair with G's, and there are no A's and T's, so we don't have to worry about that. That's lucky. Okay, so there's a double-stranded piece of DNA. So this protein, whatever enzyme it is, did anybody figure it out, what the name of the enzyme is that binds this sequence? Is it what?
In the video, you said it was E. coli or something.
So it's from a bacterium, so we'll go through that. This huge protein has a part of it that will recognize that DNA sequence. And then, when that protein binds, it cuts the double helix. So it breaks it in two, basically. And I don't remember offhand where this enzyme cuts inside that 8 nucleotide sequence, but every time that nucleotide sees that DNA sequence, it will cut the DNA. It cuts the chromosome right in half. There's something really important about that DNA sequence. It's specific to one restriction enzyme, restriction endonuclease. And that is that this is a palindrome. Does anybody see the palindrome? What is a palindrome? Somebody give an example of a palindrome in English.
Race car.
I've never heard that one. Okay, race car. So what, okay, that's good because now I'm learning something. I was going to go for a man, a plan, a canal, Panama, which is I think the longest palindrome. But so race car, what is a palindrome?
It's the same forward as back.
It's something that reads the same in one direction as it does if you turned the letters around and read it in the other direction. It's a mirror image of itself around some vertical axis. The RACE-CAR. This restriction enzyme site is a palindrome. Why is that? This is important. This is what's going to help us get on to the Google Doc assignment, which is the next thing. Which direction do we read DNA sequences in? 5' to 3'. So let's label this. The top strand is going to go 5' to 3'. So which orientation is the bottom strand? What's on the right side of the bottom strand? 5'. DNA is anti-parallel. So every time there's a 3' end, right base paired to it is a 5' end. Okay, so if you read these in 5' to 3' orientation, this is G, C, G, G, C, C, G, C. If you turn around and read it in the other direction, it's the same. You see that? G, C, G, G, C, C, G, C. So the 5' ends are the same, the 3' ends are the same. So it's not that that one strand is a palindrome. That's not true. It's that the two strands are palindromes relative to each other. They read the same on both strands, reading from 5' to 3'. That I'm pointing out to you and spending an awfully long time doing so because this is a function, this is a feature of most, if not all, restriction enzymes, of which there are thousands. Almost all of them, if not all of them, I always hedge my bets a little bit because there's almost never a sure thing in biology, at least almost all of them will recognize a palindromic DNA sequence. So if you're going to look for restriction enzyme sites, which is what this assignment next is all about, you need to know that you need to be looking for sequences of DNA that are these palindromes. They're the same sequence on the top strand as they are reading the other direction on the bottom strand. So palindrome, important topic for understanding how restriction enzymes work. That's how they know where to bind DNA and where to cut. And in this particular case, let me showcase how I would figure out what DNA sequence that thing binds. Let me see, what was it? GCGG. So if I know the first, I'm just going to search. Hopefully this is actually in a search menu in my web browser. So if you know the first 4 nucleotides of a palindrome, then you know the others because they have to be the complement, so that'd be CC, GC. So if I just search for those 8 nucleotides that I said, what bacterium does recognize that sequence, then I get all of these hits. The one that I'm looking for, just so you know, because you'll need this in probably future classes as well as this class, so that's a published article from our favorite NCBI, National Center for Biotechnology Information, the National Library of Medicine, that's PubMed, which we talked about last class. So somebody's published an article on this, another article from PubMed, another article, and then a store, New England Biolabs, that sells restriction enzymes. So they're my favorite source for information on restriction enzymes. So if you ever want to know anything about restriction enzymes, go to New England Biolabs, neb.com. Okay. And so Google works. Not one is the name of the enzyme. It recognizes the sequence GCGGCCGC. So once again, Google saves everything. So that's the sequence that this enzyme recognizes. And those dark triangles there around the sequence shows where in each of the strands of DNA this enzyme cuts. So this enzyme, interestingly, makes a staggered cut. So it cuts that strand there, and it cuts that strand there. So it makes a staggered cut. It's not a clean cut right through both strands of the DNA double helix at the same point. And for those of you that really wanted to know, this is what sticky ends are when the cut isn't blunt. When it's a staggered cut like this, this creates something called sticky ends that we may talk about at some point in the future. So that's how I figured out what that DNA sequence, what enzyme recognizes that DNA sequence. One other point that's important, this term isoschizomers, it's a mouthful. That means that there are other enzymes that will recognize this DNA sequence. So it's not just, you could buy not one from NEB if you wanted to, or you could buy something else. I'm not actually sure if this enzyme really does have an isoschizomer. Oh, there are. So there are 2 enzymes, CCI-N1 and NOT1, that both recognize the same DNA sequence. So if you see something that says it's an isoschizomer sequence, this means it's a DNA sequence that multiple different enzymes recognize. So last thing to answer that question, where did this enzyme come from? This gets extracted from a bacterium, usually. Bacteria are the only organisms that have these enzymes. And I'm not sure that this webpage is going to tell us, but if you Google NOT1, you can find out what enzyme, which bacterium it's from. But apparently not this webpage. It's not really important which bacterium this came from. I just wanted you to know, the reason for asking that question was the name of the enzyme. Does anybody else know a common restriction enzyme name? You probably don't, but you might work with these in a laboratory setting at some point. TAC. So TAC?
I'm not sure, it just sounds really familiar.
So TAC is an enzyme, and most enzymes are named in the same manner. So TAC is named in the same manner as all of these enzymes. It turns out the convention is the first letter comes from the first letter of the genus name of the bacterium. And the next two letters are the first two letters of the species name. So I think, if I remember correctly, N stands for nocardium, and OT stands for otiditis. So nocardium otiditis is the scientific name of the bacterium that this enzyme came from. So what would, let's see, someone from the back half of the class, don't make me call names or point to people, but if there was a restriction enzyme isolated from E. coli, what would those enzymes be called? Eco something, ECO, E. coli, ECO, whatever. And there are a number, those are some of the most famous restriction enzymes. I've got some in my lab. If you want to come see it, I'll show you the tiny little vial of clear liquid that's got enzymes in it. But yeah, ECO R1, ECO R2, or ECO R5, there are a bunch of different of these restriction enzymes. So those are the key points. How to look for a restriction enzyme, look for a palindromic sequence, and just know how they're named. Don't really need to be able to look up what species they're from. I won't ask you that question. Oh, and to conclude, the not one restriction site, CGC, GCGG, CCGC, I can't even read. We just talked about isochores. So where are you more likely to find, if you were going to try to cut human chromosome 14 in little bits, and you were employing not one, the enzyme that recognizes that restriction site, where on chromosome 14 is it going to cut? Where are there more G's and C's?
Right at the very beginning.
The edges of the chromosomes. This restriction site, the DNA sequence, the 8 nucleotides this enzyme is looking for, is all G's and C's. So where are you more likely to find a stretch of 8 G's and C's, let alone this particular stretch? It's going to be areas of the chromosomes that have more G's and C's. And this comes in useful too when you're trying to do something like cut up a chromosome, but you want to save the telomeres, for example. You don't want to cut them, but you want to chop up the middle half. You would look for an enzyme that recognizes what sort of a sequence you're trying to cut in here.
I have A's and T's.
You want to find something that had all A's and T's. Tell you what, it's always slower but more fun than usual. You've all got devices. Make up a palindrome that's got 6 A's and T's in it. So total 6 nucleotides long. So it should be 1, 2, 3, 4, 5, 6. But it has to read, those 6 have to read the same in that direction on the top strand and in the reverse on the bottom strand. And when you're done coming up with that, check with a neighbor and make sure it is actually a palindrome, and Google it. Just type that 6-letter sequence in to a search field on a web browser. It doesn't have to be Google. I'm biased. See if you come up with a New England Biolabs and NEB hit. Find out what restriction enzyme recognizes that palindrome you created. And I'm going to ask this as a Google Classroom question, so when you're done with all of that, then go to Google Classroom and put the information in. Paste in your palindrome and what restriction enzyme recognizes it. I will definitely circulate. If you have any questions about that, do raise your hand. I will come around and answer. I'm going to pause for a second and give some feedback. Keep working on it if you want to. Please pay attention while you work on it. I'm seeing a lot of, which is totally valuable and totally valid and the easiest one to do. I'm seeing a lot of that because it's the same T, 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 A. There's also a lot of TA, TA, TA with that in reverse, right? It's a pattern, of course. If it's a palindrome, there's going to be a pattern usually. And let's see, according to you, many of you, TTTAAA is recognized by draw 1. And TATATA is what? Did one of you pick that one? Oh, it looks like almost everybody chose draw 1. Good job, everybody. Okay, there might not be one. There might not be a restriction enzyme. I will look that up after class in case there is something weird about that sequence. In fact, there is something weird about that sequence. No, so, last point, which is something we'll get to, well, we would have gotten to later in the semester, but some of you were asking about, was this supposed to be 6 nucleotides or 12 nucleotides? I talked to a few people that asked that question. There's a reason that shorter is better for this exercise. And I'm just going to tell you simply now, we're going to move on to the last thing for the day, is that the longer the DNA sequence is, the more rare it is in terms of the sequence of our chromosomes. If that doesn't make sense, that's fine. But, well, let me put it this way. If I looked at 10 nucleotides of DNA, random 4 letters, 10 long, how many A's, if I look at 10 nucleotides, how many A's should I see on average? 2, 2.5 would be the actual average. 10 nucleotides, there are 4 nucleotides. So there should be an A every 4th nucleotide. It should be a 25% A, 25% T, G, and C, and so forth. So now, in that same 10 nucleotides, what's the likelihood, not actual probability, but just qualitatively, your guess? How likely am I to find an A next to a T? So 5' to 3' AT. Is it more likely or less likely than just finding an A?
Less likely.
Because an A can be, if there's an A, that's 1 nucleotide in every 4. Every 4th nucleotide on average should be an A. The probability of finding an A next to a T is lower. Because what else could an A be next to? Or G or C. So it's a little bit rare to find an A next to a T because every time you see an A, it could also be next to a G or next to a C. So what if I look for the specific sequence ATC? Is that more or less rare?
More rare.
Yeah, sorry. That was a horrible way to ask that question. Is it rarer or more common?
Rare.
But less likely that if you look at any one spot on a chromosome, you're going to find an A next to a T next to a C. So what if I look for ATCGAT, which is a palindrome? That 6 nucleotide sequence is pretty rare. The longer you make the DNA sequence of a restriction enzyme, the longer the identification sequence of that enzyme is, the less number of those spots in the genome there are for that enzyme to find. A 12 nucleotide sequence, a specific sequence of 12 nucleotides, if you were going to do that with this, is really uncommon throughout all of your chromosome sequences. So it's very unlikely then, in other words, you would find a restriction enzyme that even recognized a 12 nucleotide sequence. It's too rare. The enzymes don't actually exist to find DNA sequences that are that specific. So the last thing for the day is I'm going to start a Socrative exit quiz, please. Not a quiz. I'm just getting feedback. And after you're done with that, I haven't quite launched it yet. Give me just 1 minute. After you're done with that, then we're done for the day, and we'll see you next time. Which one do I want? Okay. So quick feedback from you, please. Here you go. So I've got about 3 minutes left before I have to go run teach my next class. If anybody has a question while you're doing this, feel free to come up as I'm breaking down. Got any questions at all? Come on up. You're not getting scored on whether or not you took notes. I'm just curious. How prevalent the note-taking is in the class.
Remind you, I will be here
Yeah.
Friday.
Yes. Safe travels.
Thank you.
Yeah.
I have more of those trips that are going to be coming up. Do you want me to just email you like a calendar list, or do you want like this date and then like an explanation of what it is, or?
Well, I don't mind if you're missing or not. If you want to tell me, that's totally fine. But the only time I would be really concerned about it is on exam day. So as long as we coordinate that, then it's all good.
Okay, sounds good.
Yeah.
Thanks.
Sure. Thanks for checking.